Bismillah Rahman Rahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Today, in this video lecture, we will start the MCQs from 26 because in the previous videos lecture, we already end the MCQs from 1st to 25. This is actually the video lecture that is regarded to the chapter number 3 MCQs. We already talked about the first 25 MCQs. If you missed, so go back to my channel and watch the video related to this. We also talked about the two chapter MCQs as well. It is also present into my channel. You can also watch this. Okay, now let's start the chapter number 3 MCQs with 26 number. So 26 number MCQ says that isolation is what? So isolation is actually separation of a single colony. C is the correct option. The condition required for the autoclave. So for autoclave, the condition is almost 121. This is wrong because here 15 minute is important. 120 temperature, this is also wrong. 150, 130. Okay, 150 is the correct. Why? Because in this case the microorganisms are also die. So we can say that C is also the correct option. Okay, don't be confused. 121 centigrade, we know that 121 centigrade pressure is also fine, but 20 minute in some cases it's okay, but in mostly in the cases we require 15 minutes. So we can say this is not the correct option. So the correct option is C. Okay, lysozyme is effect against, so it is mostly effective against gram-positive bacteria. B is the correct option. What is blood agar? So remember guys, blood agar is actually enriched media. It means that every type of the microorganism can be easily grow on blood agar because it contains huge amount of the nutrients. Now then we have 30 number MCQs. Infrared radiation is a method of what? It is actually the sterilization method that is mostly used to kill all type of the microorganism, and the correct option for this is mechanical method. D is the correct option. So it is actually the mechanical method. Okay, MCQs 31 says that lipolization means, so remember lipolization means freezing drying temperature. B is the correct option. Okay, MCQs 32 says that temperature used for hot air oven. So in hot air oven we mostly use 160 degrees Celsius for 1 hour. Okay, phenol coefficient indicates, so it mostly indicates the efficiency of disinfectant is the correct option. This is an agar method, agar plate method, and is commonly used for the estimation of the number of the bacteria. So we mostly use standard plate techniques is the correct option, which is also called a SPC. What is how the agar is obtained? So if you are using algae, so using which type of algae you can easily obtain the agar? So using red algae we can easily obtain the agar. B is the correct option. A gram-positive organism which produce swarming on culture media is called proteus. D is the correct option. Okay, swarming. Now enhancement of the virulence in bacteria is known as, so we also call it exhalation. C is the correct option. For effective sterilization in an autoclave, the temperature is what? So mostly the temperature is 120 centigrade. C is the correct option. Spores can be killed at autoclaving, means C is the correct. Yeah, it is also killed using glutaraldehyde. So we can say that D is the correct of both B and C. Okay. MCQs number 40 says that glassware are sterilized by using autoclave, but hot air oven is the best, because if you are using autoclave, so it will just stream sterilized. Still there is a stream present, and these streams can be sometime creating contamination, so it need to be sterilized more. So using hot air oven, the glassware will be completely sterilized. So B is the correct option. Tantalization is what? Tendrilization is actually discovered by a scientist as the name indicate Tendel. So A is the correct option. Now MCQs number 42 says that virus can be cultivated in, so it's obviously cultivated in living cells. C is the correct option. By pasteurization, so mostly by pasteurization what we have done, only pathogenic can be removed. Remember, B is the correct option. The temperature required for the pasteurization, so mostly for the pasteurization, the temperature will be below 100 centigrade. So this one MCQs is again repeated. Now 45 MCQ says that in medium other than nutrients, if any substance is used in excess, the medium is what? The medium is actually enriched medium, because you are using other than nutrients, so it is also enriched medium. Example of the indicator medium, we also call the indicator medium is also called Wilson-Bailey medium. So C is the correct option. Example of anaerobic anaerobic medium is what? The example of the anaerobic medium is Robertson-Cooked meat medium is the correct option. MCQs number 48 says that the differentiate between lactose fermented and non-lactose fermented medium is Macon-Kieger is mostly lactose fermented and non-lactose fermented, so D is the correct option. MCQs number 49 says that the best method for getting pure culture is, so agar slant is also used, streak, so C is the correct option, both A and B. MCQs 50 is the last but not the least, says that to transfer cultures from one place to another place, that device is called as what? So we mostly use to transfer one medium to another medium. Transfer culture medium from one place to another place, the device is mostly called as inoculated loop. C is the correct option. So that's all about the MCQs, 50 MCQs of the chapter number 3. Inshallah, in the next video we will start from the 51 MCQs till to 75. Okay, so that's all about the MCQs of the chapter number 3. I hope this videos help you. Thank you for watching.